Hello, everyone. Today I'll talk a little bit about the evolution of sexual dimorphism. Hello, everyone. Today I'll talk a little bit about the evolution of sexual dimorphism. Sexual dimorphism is usually explained by two main mechanisms. One of them is agonistic interactions, in which extravagant individuals, usually males, provide cues to their rivals. They have a greater fighting capacity, and so weaker rivals can use these cues to avoid initiating confrontation. The cue can be, for example, Bruno's greatest body size, which could influence Popeye to give up the fight. The other one is mate choice, in which individuals, usually females, choose their mates based on extravagant characteristics, because they can gain indirect or direct benefits from copulation. Despite the knowledge that both mechanisms are important, sexual dimorphism can be studied with this cue to one of them. That is problematic because the evolutionary direction predicted by male-male interactions may sometimes differ from the direction predicted by female mate choice. For example, the winner of a contest might be less preferred by the females. Or maybe the females might be unable to choose their partners due to male harassment. Odonata is one of these groups in which the sex role of male coloration is usually studied as important in male-male fight. But since male coloration can be a cue for individual quality, like a greater quantity of body fat or a better immune system, it would make sense for the females to choose their mates, as she will gain a chance of a better offspring. So the guiding question of the study was, does male color affect his mating chances? Our biological model was the two damselflies, Argia hasemani and Argia, in which the males are blue and the females are brownish. We hypothesized that males with a more intense coloration would present a higher copulation success. We performed our study in Brazil, more specifically in a region called Chapada Diamantina in the state of Bahia. We collected each species in different but close streams, and to investigate our hypothesis, we marked the males of both species, observed their interactions in the field, and registered which males copulated for 3 days. If the male was seen in copulation for at least one of the days, we consider him to have had reproductive success. And if he wasn't seen in copulation, we consider that he did not have reproductive success. After our observation period, we marked them and estimated male's coloration according to the female's visual system, using a spectrophotometer. To infer how females visualize the correlation of male thorax, we estimated the absolute amount of light captured by the photoreceptors, also known as KI or quantum catch. For Argia hasemani and Argia, this data was not available, so we used the Ashnura tenorística model, which presumed 3 quantum catches, 360 nanometers, 450, and 525. And what did we find? In Crassipenis, we have the chance of copulation in the y-axis and the 360 wavelength in the x-axis. And our prediction was to find a line somewhat like this. What we actually saw was a very weak or no effective color in the chance of copulation. And the same was seen for the other quantum catches. And what does that mean? Maybe the females copulate quickly when arriving at the stream without choosing, and so acquire male's protection. Or maybe male harassment enable females to choose their partners. As for Argia hasemani, we have again the same graph, chance of copulation in the y-axis and the 360 wavelength in the x-axis, and the same prediction as before. And here we found the opposite of our prediction. Though color males had a higher chance of copulation, and the same was seen for the other quantum catches. And what does that mean? This might be because females choose less colorful males, because they are less visible to predators. And by doing so, should also be protecting herself, since their copulation lasts for hours and leaves them vulnerable. To investigate this a little further, we investigated the differences between the two fields in which we found Argia hasemani and Argia, because Argia hasemani was collected in a stream with a high solar higher solar incidence, as for Clara Pidner was collected in a stream with a slightly shadowed field. So we hypothesized that males of Argia hasemani were more visible to predators than Argia crassipens, and so female mate choice might reflect this higher predation pressure. To investigate this, we model male coloration in the passerines visual spectrum, because they're important predators for both species. And this model, we use the quantum catch for passerine cones, which refer to the wavelengths 372, 453, 539, and 607. And what did we find? In the y-axis, we have the predator's quantum catch, which means how visible males are to them. And in the y-axis, we have the female's quantum catch, which means how visible the males are to them. We analyzed the 607 and 360 wavelengths because this pair showed the lowest correlation rate. Our prediction was to see both species as the wavelength in the female's visual spectrum increased, the predator's quantum catch would also increase. But Argia hasemani would be more visible than Argia crassipens due to the difference in the field. And our prediction was confirmed. Both species became more visible as the 360 wavelength increased. But also it's important to note that Argia hasemani males are disproportionately unpreferred by the females and also disproportionately more visible to predators. And we can see this difference by difference of inclination of the lines as a reflection. And what does that mean? That means that females might be choosy or not so choosy, depending on the pressure of their environment. And the final messages of our work are: female mate choice is not the main mechanism of maintenance of sexual dimorphism. However, it is still unclear why male color would affect the victory of contests, but not translate into a higher reproductive success. And finally, the intersexual and intersexual processes may act in intricate ways to determine the evolution of sexual dimorphism. And that's all. Thank you.